So, here's something worth thinking about as you settle in tonight. Right now, underneath you, beneath the mattress, beneath the floor, beneath the foundation of whatever building you're in, there's rock. And beneath that rock, there's more rock. And beneath that, more still, going down and down for thousands of miles until you reach the center of the Earth, where the temperature is roughly the same as the surface of the sun. You're lying on top of all of that right now. This thin, cool crust of a planet that is, on the inside, absolutely molten and raging and alive. And the thing is, the ground feels so solid, so permanent, so still. But it's not. It never has been. The Earth beneath you is moving right now, has been moving for billions of years. And the story of how it got here, how all of this got here, is honestly one of the most staggering things human beings have ever managed to piece together. So let's talk about it. Let's go all the way back. Not to the dinosaurs, not to the ice ages, not even to the first living thing. Let's go back to before there was an Earth at all. Let's start at the very beginning, or at least as close to the beginning as we can get, and just walk through it slowly, one piece at a time. About 4.5 billion years ago. And that number is so large it almost stops meaning anything. So let's just sit with it for a second. 4.54 billion years, give or take about 50 million. That's the age of the Earth. We know this with remarkable precision, actually, and the way we know it is itself a beautiful story. But we'll get to that. For now, just hold on to that number. 4.5 billion years. Before the Earth existed, there was a cloud, a vast, diffuse cloud of gas and dust, hanging in space, mostly hydrogen and helium left over from the Big Bang, but seasoned, and I love this word for it, seasoned with heavier elements that had been forged inside earlier generations of stars. Carbon, oxygen, silicon, iron, all of it cooked up in the nuclear furnaces of stars that had already lived and died before our sun was even born. Some of those stars died violently, in supernovae, and when they exploded, they scattered those heavy elements out into space, where they mixed with the existing gas and dust, and just drifted. For millions of years, this cloud just hung there in the Milky Way, doing nothing particularly dramatic. And then something disturbed it. Maybe a nearby supernova sent a shock wave rippling through the cloud. Maybe it was just the slow, inevitable pull of gravity finally winning out over the gas pressure holding the cloud apart. Whatever the trigger was, parts of the cloud began to collapse inward. And here's the thing about gravity. Once collapse starts, it accelerates. The denser the cloud gets, the stronger the gravitational pull, which draws in more material, which makes it denser still. It's a runaway process. And as all that gas and dust fell inward, it began to spin. Not because something was spinning it, but because of conservation of angular momentum. The same reason a figure skater spins faster when they pull their arms in. The cloud had some tiny, almost imperceptible rotation to begin with, and as it contracted, that rotation sped up. What you end up with is a flattened, spinning disk of material with a dense, hot knot at the center. That central knot is the proto-sun, getting hotter and denser by the day as more material piles on. And the disk around it? That's the solar nebula. That's where the planets are going to come from. That's where the Earth is going to come from. But not yet. Right now, it's just dust and gas, swirling around a baby star that hasn't even ignited yet. Now, here's where the engineering of planet building gets really interesting. And I mean that literally. There's a real mechanical process here, and for a long time, scientists struggled to figure out how you get from a disk of microscopic dust particles to a rocky planet 8,000 miles across. Think about that problem for a second. You've got grains of dust smaller than the width of a human hair, floating in space, and somehow they need to assemble themselves into a world. How? The first step is the easiest to understand. Dust grains collide and stick together. At that tiny scale, electrostatic forces, basically the same static cling that makes your socks stick together in the dryer, are enough to hold particles together when they bump into each other. So you get little fluffy clumps, millimeter-sized aggregates, then centimeter-sized ones. This part works fine in both theory and in laboratory experiments. Scientists have actually done this in microgravity environments, watching tiny dust grains collide and form little clusters. No problem. But then you hit what planetary scientists call the meter-size barrier. And this is genuinely one of those problems that kept researchers up at night for decades. See, once your clumps get to about a meter across, roughly the size of a boulder, things go wrong. At that size, they're too big for electrostatic forces to hold them together effectively, but they're too small for gravity to matter. And when meter-sized objects collide at the speeds typical in the solar nebula, they don't stick. They shatter. They break apart. It's like trying to build a sandcastle by throwing handfuls of wet sand at it as hard as you can. So how do you get past this? How do you go from boulders to something the size of a mountain, and then from a mountain to something the size of a planet? The current best answer involves a couple of clever mechanisms. One is called streaming instability, and it was proposed by Anders Johansson and Andrew Uden in the mid-2000s. The idea is that the dust particles in the disk interact with the gas in a way that causes them to clump together in dense filaments, almost like traffic jams on a highway. And once you get enough material concentrated in one spot, gravity takes over, and the whole clump collapses into a much larger body, maybe a kilometer across or more, essentially skipping right past the troublesome meter-size range. It's an elegant solution, and computer simulations support it beautifully. Once you've got objects a kilometer across, these are called planetesimals, the game changes completely. Now gravity is a real player. These kilometer-sized bodies attract each other, collide, merge, grow. The bigger ones grow faster because they have stronger gravity, which means they sweep up more material, which makes them bigger still. It's another runaway process, and the biggest objects in a given region of the disk start to dominate. These are the planetary embryos, and within maybe 100,000 years, which sounds like a long time but is genuinely fast in astronomical terms, you can go from a swarm of planetesimals to a handful of Mars-sized bodies orbiting the young sun. And this is where the solar system starts to look like a demolition derby. These Mars-sized embryos are on crossing orbits, and they collide with each other in impacts of almost unimaginable violence. We're talking about objects thousands of miles across, slamming together at speeds of several miles per second. The energy released in a single one of these collisions is enough to melt both objects completely, turning them into a glowing ball of magma thousands of degrees hot. And it's through these giant impacts that the final planets are assembled. Earth was built through violence, collision after collision after collision, each one adding mass, each one releasing tremendous heat, each one reshaping the growing world. Now, the most famous of these giant impacts, and one of the most dramatic events in the entire history of the solar system, is the one that gave us the moon. This is called the giant impact hypothesis, and it was first seriously proposed in the mid-1970s, though the idea had been floating around in various forms before that. The story goes like this. Roughly 30 to 50 million years after the solar system began to form, so maybe 4.5 billion years ago, the young Earth, already mostly assembled, already differentiated into a core and mantle, already a real planet, was struck by another planetary body roughly the size of Mars. This impactor has been given a name, Theia, after the Greek Titan who was the mother of Selene, the moon goddess. And when Theia hit the Earth, the result was catastrophic in the most literal sense. The collision would have been oblique, a glancing blow rather than head-on, and the energy released would have been enough to partially vaporize both bodies. Imagine the entire surface of the Earth liquefied, a magma ocean thousands of miles deep, with temperatures reaching perhaps 10,000 degrees. A huge plume of vaporized rock and debris was blasted into orbit around the Earth, forming a disk of superheated material that, over the course of perhaps just a few decades or centuries, coalesced into the Moon. Think about that timeline. The Moon may have assembled itself in less than a century, from catastrophe to companion in a human lifetime, if any humans had been around to see it. The evidence for this is actually quite compelling. When the Apollo astronauts brought back lunar samples in the late 1960s and early 70s, scientists found something surprising. The Moon's composition is remarkably similar to Earth's mantle, the rocky layer between the crust and the core. The oxygen isotope ratios are nearly identical, which is a kind of chemical fingerprint that's very hard to explain unless the Moon formed from Earth material. But the Moon is also depleted in iron compared to the Earth, which makes sense if it formed mostly from the outer, rocky layers that were blasted off during the impact, while most of the iron from both Theia and the Earth sank into Earth's core. There are still debates about the details. Some models suggest the impact was more energetic than originally thought, perhaps even vaporizing a large portion of the Earth and creating a structure called a synestia, essentially a giant, donut-shaped cloud of vaporized rock that the Moon condensed out of. Robin Canup and others have done extraordinary computational work simulating these impacts, running thousands of scenarios to see which ones produce a moon with the right mass, the right composition, the right orbital characteristics, and the models keep getting better, keep matching the data more closely. But the broad strokes have held up for nearly 50 years now. The Moon is a child of catastrophe. And here's what's really worth sitting with for a moment. After that impact, the Earth was not a place you would recognize. There was no solid surface. The entire planet was a sphere of glowing magma, radiating heat into space, with the newly formed Moon hanging enormous in the sky. Much closer than it is today, maybe only about 15,000 miles away, compared to the roughly 240,000 miles that separate us now. The sky would have been a hellscape of vaporized rock and outgassed volatiles. And this is the starting point. This is what the Earth had to cool down from, had to recover from, before anything else could happen. Before oceans, before continents, before life, before everything. The cooling happened faster than you might expect, actually. The surface of a magma ocean radiates heat very efficiently, and within maybe 1 million years, possibly even less, a thin, dark crust would have begun to form on the surface, like the skin on a cup of hot cocoa. Fragile, constantly broken and reformed as convection currents in the magma beneath churned and roiled, but there, a beginning. The first solid ground. And beneath that thin crust, something crucial was happening. The Earth was differentiating, separating into layers based on density. Iron and nickel, being the heaviest elements present in large quantities, sank toward the center, forming the core. Lighter silicate minerals floated upward, forming the mantle. This process released even more heat. Gravitational potential energy converted to thermal energy as the heavy stuff fell inward. The formation of the core alone may have raised the temperature of the Earth by another couple thousand degrees. But it also gave the Earth something extraordinary, something that would prove essential for everything that followed. A liquid iron core, churning and convecting, generating through its motion a global magnetic field. That magnetic field. Let's talk about why it matters so much, because it's one of those things that doesn't seem like a big deal until you realize what happens without it. Mars is the cautionary tale here. Mars almost certainly had a magnetic field early in its history. There's evidence of remnant magnetism frozen into ancient Martian rocks, detected by the Mars Global Surveyor spacecraft back in the late 1990s. Strong magnetic anomalies, particularly in the southern highlands, the oldest terrain on Mars. But at some point, probably around 3.5 to 4 billion years ago, Mars's core cooled enough, or at least the convection in its core slowed enough, that the dynamo sputtered out. The magnetic field collapsed, and without that protective magnetic shield, the solar wind, that constant stream of charged particles blasting outward from the sun, began stripping away Mars's atmosphere, molecule by molecule, ion by ion. Over hundreds of millions of years, Mars went from a world that probably had a thicker atmosphere, possibly liquid water on its surface, to the cold, thin-air desert we see today. Its atmospheric pressure dropped to less than 1% of Earth's. The water either froze into the ground or was lost to space. Mars dried out. Mars died, in a sense. And the difference, one of the key differences, was that magnetic field. Earth kept its dynamo running. 4.5 billion years and counting, the liquid outer core is still convecting, still generating that invisible magnetic cocoon that extends tens of thousands of miles into space, deflecting the solar wind around us, like water flowing around a stone in a stream. You've seen its effects without maybe realizing it. The aurora borealis, the northern lights, those shimmering curtains of green and purple light. That's what happens when charged particles from the sun do manage to sneak in along the magnetic field lines near the poles and collide with atmospheric gases. It's beautiful, but it's also a reminder that there's a constant bombardment happening, and the magnetic field is what keeps it from reaching the surface and gradually eroding the atmosphere that we need to breathe. So picture the Earth now, maybe 4.4 billion years ago. The surface has cooled enough to form that fragile crust. There's a magnetic field in place. And something else is happening. Something that for a long time geologists thought couldn't have happened this early, but the evidence now suggests it did. Water was beginning to accumulate on the surface. Liquid water. And this is where a really fascinating piece of evidence comes in, from a place in Western Australia called the Jack Hills. The Jack Hills are in the Narrier Gneiss terrain, which is a region of extremely ancient rocks in the Yilgarn Craton. And within those rocks, there are tiny crystals of a mineral called zircon, zirconium silicate. Zircons are remarkable. They're incredibly durable, chemically resistant, and they incorporate trace amounts of uranium when they form, which decays to lead at a known rate, giving you a built-in clock. Some of the Jack Hills zircons have been dated to 4.4 billion years old, making them the oldest known terrestrial materials. Older than any rock, because the rocks they originally formed in have long since been destroyed and recycled by geological processes. But the zircon survived, like tiny, indestructible time capsules, eventually ending up embedded in much younger sedimentary rocks. Here's what's really clever about what geologists have done with these zircons. A researcher named John Valley at the University of Wisconsin and his colleagues analyzed the oxygen isotope ratios in these ancient crystals. Now, Oxygen comes in different isotopes. Oxygen-16 is the most common, but there's also oxygen-18, which is a bit heavier. The ratio between these isotopes in a mineral tells you something about the temperature and conditions under which that mineral formed. And the oxygen isotope signatures in the Jack Hill's zircons suggest, and this was genuinely surprising when it was first published, that these crystals formed in the presence of liquid water 4.4 billion years ago. That's only about 150 million years after the Earth itself formed. The implication is that the Earth had cooled enough and had accumulated enough water to have at least some liquid water on its surface astonishingly early. Now, where did that water come from? This is one of those questions that sounds simple but has generated decades of debate. The traditional picture was that Earth formed relatively dry. The inner solar system was too hot during planet formation for water ice to be stable, so the water must have been delivered later, by comets or water-rich asteroids from the outer solar system during a period of heavy bombardment. And there's definitely something to that. But more recent work has complicated the picture. Measurements of the hydrogen isotope ratios in Earth's ocean water, specifically the ratio of regular hydrogen to deuterium, which is hydrogen with an extra neutron, those ratios are actually a closer match to certain types of carbonaceous chondrite meteorites than to most comets that have been measured. So it seems like a significant fraction of Earth's water may have come from asteroidal material, rocky bodies that formed in the outer part of the asteroid belt, where it was cool enough for hydrated minerals to exist. Water locked into the crystal structure of rocks, released when those rocks were heated during impacts or volcanic activity. And some researchers have argued that the Earth may have actually accreted with a fair amount of water from the very beginning, trapped in minerals, dissolved in the magma ocean, gradually outgassed to the surface through volcanic activity as the planet cooled. The truth is probably some combination of all of these sources. The point is, by 4.4 billion years ago, give or take, there was liquid water. Maybe not vast oceans yet, but pools, shallow seas, enough to leave its chemical fingerprint in those tiny zircon crystals. And this brings us to one of the most dramatic periods in Earth's early history, the late heavy bombardment, sometimes called the lunar cataclysm. This is a period roughly between 4.1 and 3.8 billion years ago, when the inner solar system appears to have experienced a massive spike in impact rates. The evidence comes primarily from the moon, because the moon preserves its craters beautifully. No weather, no plate tectonics, no erosion to erase them. When Apollo astronauts brought back samples from the large lunar basins, those enormous dark patches you can see on the moon's face, the Maria, radiometric dating of the impact melt rocks clustered heavily around 3.9 billion years ago. Basin after basin, same rough age. It seemed like the moon had been absolutely pummeled during this narrow window of time. And if the moon was getting hit, Earth was getting hit even harder. Earth is a bigger target with stronger gravity. What could have caused such a sudden surge in impacts? Hundreds of millions of years after the planets had already formed, and the solar system should have been settling down. This is where a really elegant model comes in, developed in the early 2000s by a group of researchers working in Nice, France, so it's called the NICE model, pronounced like the city. The key figures include Rodney Gomes, Hal Levison, Alessandro Morbidelli, and Cleomenes Tsiganis. What they proposed was that the giant planets, Jupiter, Saturn, Uranus, and Neptune, weren't always in the orbits they occupy today. Early on, they were in a more compact configuration, closer together and closer to the Sun. Beyond them lay a massive disk of icy planetesimals, leftover building blocks that never got incorporated into a planet. Over time, gravitational interactions between the giant planets in this disk caused the planets' orbits to slowly migrate. And then, at some point, Jupiter and Saturn crossed a critical orbital resonance. Their orbital periods hit a ratio of 2:1, meaning Jupiter completed exactly 2 orbits for every one of Saturn's. When that happened, it was like a switch flipping. The resonance pumped up the eccentricities of the giant planets' orbits, destabilized the whole system. Uranus and Neptune were flung outward into the planetesimal disk, scattering icy bodies everywhere, some inward toward the inner solar system, some outward to form the Kuiper belt. The result was a rain of impactors throughout the solar system, lasting maybe 100 to 200 million years. Now I should say that the late heavy bombardment as a sharp spike, a true cataclysm, has become more controversial in recent years. Some researchers looking at the lunar data more carefully have argued that the clustering of ages around 3.9 billion years might be a sampling bias, because most Apollo samples came from a limited number of sites, many of which could have been contaminated by ejecta from a single enormous impact, the one that created the Imbrium Basin. So maybe there wasn't a sudden spike at all. Maybe it was more of a gradual decline in bombardment rates from the very beginning, a long tail of impacts that slowly tapered off. The debate is ongoing, and the truth matters a lot for understanding the conditions under which life first arose. Because here's the thing. If the late heavy bombardment really was as intense as the original model suggested, it raises a profound question about life's origins. Some of the largest impacts during this period would have been energetic enough to vaporize entire oceans, sterilizing the surface of the Earth completely. If that happened, then life either originated after the bombardment ended, after about 3.8 billion years ago, or it originated earlier, was wiped out, and then originated again. Or, and this is a fascinating possibility, life found refuge deep underground in rock fractures and hydrothermal systems, where even the most catastrophic surface impacts couldn't reach it. Extremophile microorganisms living kilometers below the surface, riding out the apocalypse in the warm, wet darkness of the deep crust. And this connects to something we should talk about, because it's one of the biggest unsolved questions in all of science: how life began in the first place, the transition from chemistry to biology, from molecules that just sit there and react according to the laws of thermodynamics, to molecules that copy themselves, that carry information, that evolve. We don't know exactly how it happened, but we have some very compelling ideas, and the geological setting of the early Earth, that hot, volcanic, chemically rich world with its young oceans and its constant energy inputs, turns out to be exactly the kind of environment where the right chemistry could have gotten very, very interesting. Think about what the early Earth actually looked like around 4 billion years ago. You've got a planet that's still radiating a lot of internal heat. The mantle is hotter, volcanism is more vigorous, and there are hydrothermal vents scattered across the ocean floor. Places where superheated water, laced with dissolved minerals and gases, comes pouring out of cracks in the crust. You've got an atmosphere that's mostly nitrogen and carbon dioxide, with traces of methane, ammonia, water vapor, hydrogen sulfide. No free oxygen. That comes much later, and when it does, it changes everything. But we'll get to that. The oceans themselves are slightly acidic, rich in dissolved iron, and they're being constantly stirred by tidal forces that are much stronger than today's, because the moon is closer. Remember, the moon formed from that giant impact with Theia, and right after formation, it was maybe 10 times closer than it is now. So the tides on early Earth were enormous. Massive surges of water washing over volcanic shorelines, flooding tidal pools, retreating, flooding again. And that rhythmic wetting and drying turns out to matter a lot for prebiotic chemistry, because certain key reactions, The kinds that build long-chain molecules from simpler precursors work better when solutions concentrate through evaporation and then get diluted again. It's like a natural laboratory running the same experiment over and over, billions of times. So here's the big question. How do you get from simple molecules, water, carbon dioxide, ammonia, methane, hydrogen cyanide, to something that can copy itself, something that carries information and passes it on? Because that's really what life is, at its most fundamental. It's chemistry that remembers, chemistry that makes more of itself. And the gap between ordinary chemistry and that kind of chemistry is enormous. It's arguably the biggest gap in our entire understanding of natural history. The classic experiment, the one everyone learns about in school, is the Miller-Urey experiment from 1953. Stanley Miller was a graduate student at the University of Chicago, working with Harold Urey, who was a Nobel laureate in chemistry, and their idea was beautifully simple. They sealed a mixture of gases, methane, ammonia, hydrogen, and water vapor, inside a glass apparatus, and they zapped it with electrical sparks to simulate lightning. Then they let the products collect in a water trap at the bottom. After just a week, the water had turned pink, and when Miller analyzed it, he found amino acids, glycine, alanine, aspartic acid, the actual building blocks of proteins, just from sparks and simple gases. It was a sensation. It seemed to suggest that the raw ingredients of life could form almost effortlessly under early Earth conditions. Now, the Miller-Urey experiment is still important, still foundational, but our understanding has gotten a lot more nuanced since 1953. For one thing, we now think the early atmosphere wasn't quite as hydrogen-rich as Miller and Urey assumed. It was probably more neutral, dominated by carbon dioxide and nitrogen rather than methane and ammonia. And when you repeat the experiment with a more realistic atmospheric composition, you still get some amino acids, but the yields are lower. So the atmosphere alone might not have been the primary factory for prebiotic molecules. But here's what's interesting. After Miller died in 2007, one of his former students, Jeffrey Beta, went back and reanalyzed the original sealed vials from the 1953 experiment using modern analytical techniques, mass spectrometry that Miller never had access to. And Bata found that the original experiment had actually produced far more amino acids than Miller ever reported, over 20 different ones, plus other organic compounds. The chemistry was even richer than anyone realized. But amino acids are just one piece of the puzzle. You also need nucleotides, the building blocks of RNA and DNA, and nucleotides are much harder to make. They're more complex molecules, each one consisting of a sugar, a phosphate group, and a nitrogenous base, all linked together in a specific way. For decades, this was considered a nearly insurmountable problem. You could make the individual pieces separately in the lab, but getting them to assemble into actual nucleotides under plausible prebiotic conditions seemed almost impossible. The sugar component, ribose, is particularly fragile. It falls apart almost as fast as it forms. And then in 2009, a chemist named John Sutherland, working at the University of Manchester, published a paper that kind of changed the whole conversation. Sutherland showed that you don't actually need to build nucleotides by assembling their three components separately and then stitching them together. Instead, he found a pathway where simpler precursor molecules, things like cyanamide, cyanoacetylene, glycolaldehyde, and phosphate, could react together in a series of steps that produce nucleotides directly without ever going through the individual sugar and base intermediates. The chemistry bypasses the fragile ribose problem entirely. And the conditions required? Ultraviolet light, wet-dry cycling, and phosphate-rich environments, all of which the early Earth had in abundance. Sutherland's work pointed toward something called the RNA world hypothesis, which is one of the leading ideas about how life got started. The basic concept is this: before there were cells, before there were proteins, before there was DNA, there was RNA. RNA is this remarkable molecule that can do double duty. It can carry genetic information, like DNA does, but it can also fold up into complex shapes and catalyze chemical reactions, like proteins do. We call these catalytic RNA molecules ribozymes, and they're real. They exist in modern cells, doing important jobs. The ribosome itself, the molecular machine that builds all the proteins in your body, is fundamentally an RNA machine. The catalytic core of the ribosome is made of RNA, not protein. That's a fossil, in a sense. A molecular fossil from the RNA world, preserved inside every living cell on Earth. So the idea is that early life was based on RNA molecules that could both store information and catalyze their own replication. No proteins needed, no DNA needed, just RNA doing everything. And over time, this RNA-based life evolved greater complexity. Proteins took over the catalytic roles because they're more versatile. DNA took over the information storage role because it's more stable. And RNA got demoted to its current role as a messenger, a middleman shuttling information from DNA to the protein-building machinery. It's a compelling narrative, and a lot of the chemistry checks out. But there are other ideas too, and they're not mutually exclusive. One of the most fascinating involves those hydrothermal vents I mentioned. Not the dramatic black smokers you might have seen in documentaries, the ones that spew superheated water at 350 degrees Celsius from volcanic ridges. Those are probably too hot and too acidic for prebiotic chemistry. Instead, the focus has shifted to a different kind of vent, called alkaline hydrothermal vents. The best-known example is a vent field called Lost City, discovered in 2000 on the Mid-Atlantic Ridge. Lost City is remarkable. The vents there aren't powered by volcanic heat. They're powered by a chemical reaction between seawater and a type of rock called peridotite, in a process called serpentinization. When seawater percolates down into the ocean crust and reacts with iron and magnesium-rich minerals, it produces heat, hydrogen gas, and highly alkaline fluids. The resulting vent structures are tall, pale, ghostly chimneys made of carbonate minerals, and they can persist for tens of thousands of years, much longer than black smokers. Now here's what's really clever about the alkaline vent hypothesis, which was championed for decades by a geochemist named Mike Russell, starting in the late 1980s. Russell noticed something striking about the way all living cells generate energy. Every cell on Earth, every bacterium, every plant cell, every cell in your body, generates energy using the same fundamental trick. They maintain a gradient of protons, hydrogen ions, across a membrane, and then they let those protons flow back through a molecular turbine called ATP synthase, which generates ATP, the universal energy currency of life. This is called chemiosmosis, and it was discovered by Peter Mitchell, who won the Nobel Prize for it in 1978. It's how your mitochondria work. It's how chloroplasts work. It's how bacteria power themselves. It's everywhere. And Russell's insight was this: alkaline hydrothermal vents naturally create exactly this kind of proton gradient. The vent fluid is alkaline, meaning it has a low concentration of protons. The surrounding ocean water, which on the early Earth was slightly acidic and rich in dissolved carbon dioxide, has a high concentration of protons. So right at the boundary where vent fluid meets ocean water, you get a natural proton gradient across the thin mineral walls of the vent's porous structure. Those mineral walls act like primitive membranes, and the gradient provides free energy. energy that could have driven the formation of organic molecules, the assembly of more complex structures, maybe even the first rudimentary metabolism. Life, in this view, didn't start with replication. It started with energy flow. It started with chemistry being pushed uphill by the natural thermodynamic disequilibrium between alkaline vent fluid and acidic ocean water. It's a beautiful idea, and it has the advantage of explaining why all life uses chemiosmosis. If life originated at alkaline vents, then chemiosmosis isn't something that evolved later. It was there from the very beginning, baked into life's origin. The first living systems didn't need to invent a way to generate energy gradients. They were born inside one. But regardless of which specific setting you favor, warm tidal pools, alkaline vents, ice surfaces where concentration effects enhance reactions, or some combination, the geological evidence suggests that life got started remarkably quickly. Those Jack Hill zircons we talked about, the ones John Valley studied, tell us there was liquid water by 4.4 billion years ago. The oldest widely accepted evidence for life comes from rock formations in places like the Isua Greenstone Belt in southwestern Greenland and the Nuvvuagittuq Belt in northern Quebec. The Isua rocks are about 3.7 billion years old, and they contain carbon isotope signatures, ratios of carbon-12 to carbon-13, that look biological. Living organisms preferentially use the lighter carbon-12 isotope, so when you find rocks enriched in carbon-12, it's a fingerprint of biological activity. The Nuvvuagittuq rocks might be even older, possibly 4.3 billion years, though their age is debated. If those dates hold up, it means life appeared almost as soon as conditions allowed it, almost as soon as the surface cooled enough for liquid water to persist and the worst of the bombardment tapered off. Something started metabolizing, started replicating, started evolving. That speed is either telling us that life's origin is relatively easy, that given the right conditions, it happens almost inevitably, or it's telling us that we got extraordinarily lucky, very early. Most astrobiologists lean toward the first interpretation, which is a hopeful thought if you're wondering whether life exists elsewhere in the universe. But here's the thing about those earliest organisms. They were almost certainly simple, single-celled, and anaerobic. They didn't use oxygen because there wasn't any free oxygen to use. The early Earth's atmosphere and oceans were essentially oxygen-free, and they would remain that way for a very, very long time. For roughly the first 2 billion years of life's existence, the planet was inhabited exclusively by microorganisms, bacteria and archaea, living in an anoxic world. And during that immense stretch of time, something was quietly evolving that would eventually transform the planet more dramatically than any asteroid impact or volcanic eruption ever could. Somewhere in the shallow, sunlit waters of the Archean Ocean, a lineage of bacteria was developing a new way to harvest energy from sunlight. Not just any kind of photosynthesis, mind you. There were already bacteria doing simpler versions of that, pulling electrons from hydrogen sulfide or iron to power their chemistry. But this new trick was different. This new trick would crack water molecules apart, and in doing so, it would release a waste product that would poison most of the life on Earth and permanently alter the chemistry of the atmosphere, the oceans, and even the rocks beneath the seafloor. That waste product was oxygen. The organisms that figured this out were cyanobacteria, and you've almost certainly seen their descendants. They're the blue-green scum on the surface of a pond, the dark crust on desert rocks, the tiny green filaments waving in a stream. They look humble. They look like nothing. But cyanobacteria are arguably the most consequential organisms in the history of this planet. What they did, what they're still doing, is called oxygenic photosynthesis. And to understand why it changed everything, you need to appreciate just how chemically violent it really is. Here's the thing about water. Water is a remarkably stable molecule. Hydrogen and oxygen are bound together tightly, and splitting that bond takes serious energy. The earlier forms of photosynthesis used easier electron donors. Hydrogen sulfide, ferrous iron, things that give up their electrons without too much fuss. But water? Water doesn't want to let go. To rip electrons from water, you need a molecular machine that can generate an extraordinarily strong oxidizing potential, and cyanobacteria evolved exactly that. They developed a protein complex called photosystem II, which contains a cluster of 4 manganese atoms and 1 calcium atom arranged in a specific geometry. This little cluster, sometimes called the oxygen-evolving complex, is where the magic happens. It strips electrons from water molecules one at a time, 4 times in a row, and each time it cycles, it accumulates enough oxidizing power to split 2 water molecules into 4 protons, 4 electrons, and 1 molecule of O2. Molecular oxygen, the stuff you're breathing right now. Think about what that means. Every oxygen molecule in the air around you, every breath you take, traces back to this reaction, to this little manganese cluster inside a protein that some bacteria figured out roughly 2.5 to 3 billion years ago. Maybe earlier. The exact timing is one of the great ongoing debates in geobiology. Some researchers, like Roger Buick at the University of Washington, have argued for whiffs of oxygen appearing as early as 3 billion years ago, based on trace element signatures in ancient sediments. Others place the evolution of oxygenic photosynthesis closer to 2.7 or 2.8 billion years ago, based on molecular fossils, specifically biomarker molecules called steranes and hopanes found in ancient shales from Australia. But the biomarker evidence has been contested. Jochen Brox at the Australian National University, who originally reported some of those molecular fossils in the early 2000s, later went back and showed that several of the key samples had been contaminated by younger organic material that had seeped into the rocks. It was a remarkable act of scientific integrity, going back and essentially dismantling your own earlier discovery because the evidence demanded it. So the exact date when cyanobacteria first started splitting water remains fuzzy. But here's what we do know with much more confidence. For a long time after oxygenic photosynthesis evolved, the oxygen it produced didn't accumulate in the atmosphere, not in any meaningful way. And the reason is beautifully simple. The planet had an enormous appetite for oxygen. Think of the early Earth as a giant sponge for any free O2 that appeared. The oceans were full of dissolved ferrous iron in its reduced 2+ oxidation state. And when oxygen met that iron, it reacted immediately, oxidizing the iron to its 3+ state, which is insoluble. It precipitated out of the water as iron oxide minerals and sank to the ocean floor, layer after layer after layer. And this is where we get one of the most visually stunning rock formations in the geological record, banded iron formations, or BIFs. If you've ever seen pictures of those gorgeous red and silver striped rocks from places like the Hamersley Basin in Western Australia, or the Mesabi Range in Minnesota, or Kursk in Russia, those alternating bands of iron-rich and iron-poor sediment, you're looking at the chemical diary of a planet slowly being poisoned by its own biology. Each iron-rich band represents a period when oxygen was being produced and immediately consumed by reacting with dissolved iron. The iron-poor silica bands may represent periods when the local iron supply was temporarily exhausted or when oxygen production slowed. The banding might also reflect seasonal cycles or episodic blooms of cyanobacteria. or fluctuations in iron-rich water upwelling from the deep ocean. The exact mechanism behind the banding is still debated, but the big picture is clear. For hundreds of millions of years, oxygen was being generated and then immediately locked away in rock. The atmosphere stayed anoxic, and it wasn't just iron soaking up the oxygen. Volcanic gases, hydrogen sulfide, sulfur dioxide, methane, carbon monoxide, all of these are oxygen sinks. Every time a volcano erupted, it belched out reducing gases that would react with and consume any free oxygen in the atmosphere. So you had this long, slow tug-of-war between the biological source of oxygen and the geological sinks consuming it. And for a staggeringly long time, the sinks were winning. But here's what's really clever about the way this played out. Every time oxygen reacted with dissolved iron and the iron oxide precipitated to the seafloor, that iron was permanently removed from the ocean. It was buried, locked away in sedimentary rock, which means the ocean's capacity to absorb oxygen was slowly, irreversibly shrinking. Think of it like a bucket with a hole in the bottom. You can pour water in and it drains out. But if the hole is gradually getting smaller, eventually the water level starts to rise. That's essentially what happened. The oxygen sinks were being consumed faster than they were being replenished, and at some point, the balance tipped. That tipping point is what we call the Great Oxidation Event, and it happened roughly 2.4 billion years ago. The evidence for it comes from several independent lines of geological data, but one of the most elegant is something called mass-independent fractionation of sulfur isotopes, which sounds incredibly technical, but the concept is actually quite beautiful. In an oxygen-free atmosphere, ultraviolet light from the sun can drive photochemical reactions with sulfur dioxide gas that produce a very specific, unusual pattern in the ratios of sulfur isotopes. Sulfur-33 in particular behaves anomalously compared to what you'd expect from normal mass-dependent processes. This signal gets preserved in sulfide and sulfate minerals in ancient sedimentary rocks. And when you look at the rock record, you see this anomalous sulfur signal everywhere in rocks older than about 2.4 billion years. And then, quite abruptly, geologically speaking, it disappears. It vanishes. And it never comes back. The interpretation is straightforward. Once oxygen rose above a certain threshold in the atmosphere, probably just a few parts per million, which is still tiny compared to today's 21%, it created an ozone layer that blocked the specific wavelengths of UV light responsible for those anomalous sulfur reactions. The signal switched off like a light, and that transition, recorded in sulfur isotopes in rocks all over the world, gives us a remarkably precise marker for when oxygen first became a persistent presence in Earth's atmosphere. Now, when I say persistent presence, I want to be careful, because the Great Oxidation Event wasn't a smooth one-way ramp from 0 to 21% oxygen. It was messy. Oxygen levels probably rose, then crashed, then rose again, oscillating wildly over tens or even hundreds of millions of years. There's evidence for at least one, possibly several, dramatic glaciations right around this time. The Huronian glaciations, named after rocks near Lake Huron in Ontario, dating to roughly 2.4 to 2.1 billion years ago. And there's a compelling hypothesis linking these ice ages directly to the rise of oxygen. Here's how it works. Before the Great Oxidation Event, the atmosphere contained significant amounts of methane, CH4, produced by methanogenic archaea, those ancient microorganisms we talked about earlier. Methane is a potent greenhouse gas, much more effective at trapping heat than carbon dioxide. It was helping keep the early Earth warm despite the fact that the sun was still only about 80% as bright as it is today. That's the faint young sun problem, and methane was probably a big part of the solution. But when oxygen started accumulating, it reacted with atmospheric methane, oxidizing it to carbon dioxide and water. CO2 is a greenhouse gas too, but molecule for molecule, it's far weaker than methane. So the rise of oxygen effectively destroyed the methane greenhouse, and with the sun still relatively dim, temperatures plummeted. The result may have been the most severe glaciation in Earth's history. A Snowball Earth event, or something close to it, with ice extending to low latitudes, possibly all the way to the equator. Imagine that. Life invents a new way to harvest sunlight, and the waste product from that invention triggers a global ice age. The cyanobacteria didn't know they were doing this. Obviously, there was no intent, no plan, just chemistry and natural selection playing out over hundreds of millions of years, and the unintended consequence was a planetary catastrophe. Some researchers have called the Great Oxidation Event the first mass extinction, because for the anaerobic organisms that had dominated Earth for over 1 billion years, free oxygen was lethal. It was a corrosive, reactive poison that destroyed their enzymes, shredded their DNA, wrecked their metabolic machinery. The rise of oxygen would have been, from their perspective, an apocalypse. And yet, here's the beautiful paradox. That same poison, that corrosive waste gas, would eventually become the foundation for complex life. Because aerobic respiration, the metabolic process that you and I and every animal on Earth uses to generate energy, is extraordinarily efficient compared to anaerobic alternatives. When you burn glucose with oxygen, you extract roughly 18 times more energy per molecule than you get from fermentation. That energy surplus is what ultimately made it possible for cells to grow larger, to develop more complex internal structures, to cooperate in multicellular arrangements. Without oxygen, you almost certainly don't get animals, don't get plants, don't get fungi, don't get anything you can see with the naked eye. The cyanobacteria's waste product was the key that unlocked biological complexity. But that complexity was still a very long way off. After the Great Oxidation Event, oxygen levels didn't just keep climbing. They appear to have stabilized at a relatively low level, maybe 1 to 10% of present atmospheric levels, and then stayed there, more or less, for over 1 billion years. Geologists sometimes call this interval the boring billion, stretching from roughly 1.8 billion to about 800 million years ago. And on the surface, it does look boring. The rock record from this period shows relatively little change, no dramatic glaciations, no big shifts in ocean chemistry, no obvious bursts of biological innovation. Stable, quiet, uneventful. But boring might be the wrong word, because during this long, seemingly static interval, something profoundly important was happening at the cellular level. Somewhere in this stretch of time, and again, the exact timing is hotly debated, a small, simple cell swallowed another cell, and instead of digesting it, kept it alive inside itself. This is the endosymbiotic origin of eukaryotic cells, and it's one of the most extraordinary events in the history of life. The idea was championed most forcefully by Lynn Margulis, starting in the late 1960s, when she was a young biologist at Boston University, and her paper on the subject was rejected by roughly 15 journals before finally being published in 1967. The scientific establishment thought she was wrong. They thought she was spinning fairy tales, proposing something that sounded more like science fiction than serious biology. The idea that one cell could live permanently inside another, that the very organelles you find in every complex cell on Earth today were once free-living bacteria. It struck most of her colleagues as outlandish. But Margulis was stubborn in the best possible way, and she was right. Let's slow down and really sit with this, because the story of endosymbiosis is one of those ideas that, once you truly understand it, changes the way you look at every living thing around you. Every cell in your body, every cell in every animal, every plant, every fungus, every protist, contains structures that were once independent organisms. They had their own lives, their own evolutionary histories, their own DNA. And they still carry remnants of that independence right now, inside you, as you lie here listening to this. Start with mitochondria. You've probably heard of them. Maybe remember them from school as the powerhouses of the cell. That phrase is so overused, it's almost lost its meaning. But it's actually a pretty good description. Mitochondria are the organelles that perform aerobic respiration. They're the ones that take the food molecules your body breaks down and combine them with oxygen to produce ATP, the energy currency that powers almost everything your cells do. Without mitochondria, your cells would be stuck with fermentation, squeezing out maybe 2 ATP molecules per glucose instead of the roughly 36 that aerobic respiration delivers. You simply could not sustain a complex multicellular body on fermentation alone. The math doesn't work. Now here's what Margulis proposed, building on an idea that had actually been floating around since the early 1900s, when a Russian botanist named Konstantin Mereshkovsky first suggested it in 1905. The idea is that mitochondria were originally free-living alpha proteobacteria, small, oxygen-breathing bacteria that were exceptionally good at aerobic respiration. At some point, probably during the Boring Billion, or perhaps even earlier, an archaeal host cell, something from the domain archaea, which are prokaryotes, but genetically distinct from bacteria, engulfed one of these alpha proteobacteria. Maybe it was trying to eat it. Maybe the bacterium invaded as a parasite. We don't know the initial circumstances, but whatever happened, the bacterium wasn't digested. It survived inside the host cell. And over time, an extraordinary partnership developed. The bacterium was brilliant at extracting energy from organic molecules using oxygen. The host cell provided a stable environment, raw materials, protection. Gradually, over millions of years, the two became so dependent on each other that neither could survive alone. The bacterium lost many of its own genes for building a cell wall, for independent reproduction, for all the things a free-living organism needs, because the host cell was providing those functions. Many of those genes actually transferred from the bacterium's genome into the host cell's nuclear DNA, which is a wild thing to think about. Parts of what you consider your DNA were originally bacterial genes. Meanwhile, the bacterium retained its own small circular genome, its own ribosomes, its own double membrane. That double membrane is actually one of the key pieces of evidence for endosymbiosis, because it makes perfect sense if the inner membrane was the bacterium's original membrane, and the outer membrane came from the host cell's engulfing vesicle. Margulis laid all of this out, and the evidence she marshaled was compelling even in 1967. Mitochondria divide by binary fission, just like bacteria. They have their own DNA, and it's circular, just like bacterial DNA. Their ribosomes are more similar to bacterial ribosomes than to the ribosomes in the cytoplasm of eukaryotic cells. Certain antibiotics that target bacteria, like chloramphenicol, also inhibit mitochondrial function, which makes perfect sense if mitochondria are descended from bacteria. And when molecular phylogenetics matured in the 1980s and 90s, the case became airtight. You could build evolutionary trees using mitochondrial gene sequences and show definitively that mitochondria are nested within the alpha proteobacteria. they're not just similar to bacteria. They are bacteria. Or rather, they descend from bacteria. Margulis was vindicated, and endosymbiosis went from fringe hypothesis to textbook fact. She also proposed, and this is equally important, that chloroplasts, the organelles that perform photosynthesis in plant and algae cells, originated the same way. A eukaryotic cell that already had mitochondria engulfed a photosynthetic cyanobacterium, and again, instead of digesting it, kept it alive. The cyanobacterium became the chloroplast. Same evidence. Own DNA, own ribosomes, double membrane, binary fission, molecular phylogenetics, placing chloroplasts firmly within the cyanobacterial lineage. So when you look at a leaf, you're looking at cells that contain the descendants of two separate bacterial invasions. One that gave them the ability to breathe oxygen, and another that gave them the ability to harvest sunlight. Layers of ancient partnerships nested inside each other. Now, Margulis went further than most biologists were willing to follow. She also proposed that other cellular structures, like cilia and flagella, the whip-like appendages that some cells use to move, originated through endosymbiosis with spirochete bacteria. That particular claim has not been supported by the molecular evidence, and most biologists today don't accept it. Margulis held on to it stubbornly until her death in 2011, and it's a reminder that being spectacularly right about one thing doesn't mean you're right about everything. But on the big claims, mitochondria and chloroplasts, she was absolutely, fundamentally correct. And the story of how she fought for that idea against an entire establishment is one of the great tales in the history of biology. So, let's think about what this means for the timeline we've been following. During the Boring Billion, oxygen levels were low but present. Eukaryotic cells, cells with nuclei, internal membranes, and mitochondria, were evolving and diversifying. The oldest fossils that are widely accepted as eukaryotic are around 1.8 to 2 billion years old. There are acritarchs, tiny organic-walled microfossils from this period that are larger and more complex than anything bacteria produce. And there are fossils like Grypania spiralis, a coiled ribbon-like organism found in rocks about 2.1 billion years old from Michigan, which many paleontologists interpret as a eukaryotic alga, though that interpretation is debated. Here's the thing about the Boring Billion, though. Even with eukaryotes present, life didn't seem to be in any hurry. For over 1 billion years, you had single-celled or simple multicellular eukaryotes drifting around in oceans with modest oxygen levels, and not much seemed to change. Why? What was holding things back? One idea, and this is an area of active research, is that it was the oxygen, or rather, the lack of enough oxygen. Nick Butterfield at Cambridge has argued that the Boring Billion wasn't really boring at all, that important evolutionary innovations were accumulating, including the origins of sex, predation, and multicellularity. But other researchers, like Tim Lyons and his group at UC Riverside, have focused on ocean chemistry. During the Boring Billion, the deep oceans appear to have been largely anoxic, and in many places euxinic, meaning not just oxygen-free, but actively sulfidic, rich in hydrogen sulfide, which is toxic to most eukaryotic life. Imagine vast stretches of ocean that were essentially poisonous. The surface waters had some oxygen thanks to cyanobacteria, but below a certain depth, conditions were hostile. This would have limited the habitats available for oxygen-dependent eukaryotes and constrained how large and complex they could become. There's also the question of nutrients. Phil Planavsky and others have shown that trace metals like molybdenum and phosphorus may have been scarce during this interval, which would have limited biological productivity. If you don't have enough nutrients to fuel big algal blooms, you don't get much organic carbon burial, you don't draw down much carbon dioxide, and the whole system stays in a kind of low-energy equilibrium. Stable, yes, but also stuck. And then, around 800 million years ago, things started to change. The boring billion ended, and the Earth entered one of the most dramatic and violent periods in its entire history. This is the Neoproterozoic, and it's when everything goes haywire in the most productive possible way. The first thing that happened, or at least the most dramatic thing, was ice. Lots and lots of ice. We talked earlier about the Huronian glaciations back around 2.3 billion years ago, triggered by the collapse of the methane greenhouse after the Great Oxidation Event. Well, the Neoproterozoic brought glaciations that may have been even more extreme. These are the Snowball Earth events, and the evidence for them is genuinely astonishing. The key figure here is Paul Hoffman, a geologist at Harvard, who in the late 1990s revived and championed the Snowball Earth hypothesis, originally proposed by Joe Kirschvink at Caltech in 1992. Kirschvink had noticed something strange in the geological record from around 700 million years ago. You could find glacial deposits, tillites, dropstones, striated bedrock, at paleolatitudes that were clearly tropical, not near the poles, not at high latitudes, at the equator. Ice at the equator. Think about what that implies. If glaciers were grinding across tropical continents, then the entire planet must have been frozen. Not just an ice age like the ones in the Pleistocene, where ice sheets covered Canada and Scandinavia, but the tropics stayed warm. This was something categorically different. Ice from pole to pole. The entire ocean surface frozen over, possibly to a depth of 1 kilometer or more in places. Global average temperatures perhaps dropping to -40 or -50 degrees Celsius. Hoffman took Kirschvink's idea and built a comprehensive case for it, working extensively in Namibia, where Neoproterozoic rocks are beautifully exposed. What he found was a repeating sequence: glacial deposits sitting directly beneath thick layers of carbonate rock, limestone and dolostone, called cap carbonates. And these cap carbonates are chemically peculiar. They have unusual carbon isotope signatures and strange sedimentary structures that suggest they were deposited extremely rapidly in warm, carbon dioxide-rich waters. The sequence tells a story. The glaciation locks up the planet in ice. Volcanic activity continues beneath the ice, pumping carbon dioxide into the atmosphere. But there's no weathering to remove it, because the continents are buried under glaciers and the oceans are sealed beneath ice. Carbon dioxide builds up to extraordinary levels, maybe 300 times pre-modern levels, some models suggest, until eventually the greenhouse effect becomes so powerful that it overwhelms the ice albedo feedback and the whole system snaps. The ice melts catastrophically, perhaps in just a few thousand years. You go from a frozen world to a hothouse almost overnight in geological terms. Intense chemical weathering of newly exposed rock dumps, calcium and carbonate ions into the oceans, and the cap carbonates precipitate out in a frenzy. There were at least 2 major Snowball Earth events in the Neoproterozoic. The Sturtian glaciation, around 717 to 660 million years ago, and that's a glaciation that lasted nearly 60 million years, which is staggering to think about. And the Marinoan glaciation, from about 650 to 635 million years ago. Some researchers have proposed a third, the Gaskier's glaciation, around 580 million years ago. Though that one may have been less severe, perhaps more of a slush ball than a full snowball. And here's where the story connects back to life in a way that's almost too perfect. because it's right after these Snowball Earth events, in the warm, oxygen-rich aftermath, that we see the first unambiguous, large, complex multicellular organisms in the fossil record. The Ediacaran biota, named after the Ediacara Hills in South Australia where they were first described, appear in rocks dating to around 575 million years ago, shortly after the Marinoan glaciation ended. These are the oldest large, complex life forms we know of, and they are genuinely strange. Not strange in the way that, say, a deep-sea anglerfish is strange, something alien, but still recognizably an animal. These Ediacaran organisms are strange in a much more fundamental way. Many of them don't obviously belong to any group of life that exists today. When you look at them, you're not sure if you're looking at an animal, a fungus, a colonial organism, a giant single-celled thing, or something else entirely. Something that has no modern analog because its entire body plan was a dead end, an experiment that nature tried and then quietly abandoned. Let me paint you a picture of what some of these looked like. Imagine a frond, something that looks a bit like a fern leaf or a feather, anchored to the seafloor by a small hold-fast disc. It could be half a meter tall, maybe more. It's got a quilted texture, almost like a stitched mattress, with repeating segments that branch off a central axis. This is Charnia, one of the most famous Ediacaran fossils, and it was actually first discovered in Charnwood Forest in England in 1957 by a schoolboy named Roger Mason, though a girl named Tina Negus had spotted it a year earlier and reported it to her teacher, who didn't believe her. There's a whole story there about whose discovery it really was, and it took decades for Negus to get proper recognition. But the point is, Charnia lived on the deep sea floor, well below the photic zone where sunlight could reach. So it wasn't photosynthesizing. It was probably absorbing dissolved organic matter directly through its surface, or maybe filter feeding in some rudimentary way. And it had no mouth, no gut, no obvious internal organs of any kind. Just this beautiful, fractal, quilted structure sitting on the bottom of a dark ocean. Then there's Dickinsonia, which might be the most debated organism in all of paleontology. Picture something flat and oval, like a bath mat, with a series of rib-like segments radiating out from a central groove. Some specimens are over a meter across, but they're paper-thin. For decades, people argued about what Dickinsonia was. A jellyfish, a worm, a lichen, a colonial organism, a failed experiment in multicellular life that has no living descendants? In 2018, a team led by Ilya Bobrovsky at the Australian National University found cholesterol molecules, cholesteroids, preserved in Dickinsonia fossils from the White Sea region of Russia. And here's why that matters. Cholesterol is a biomarker specifically associated with animals, not fungi, not bacteria, not algae. Animals. So Dickinsonia was almost certainly an animal of some kind, which makes it one of the oldest animals in the fossil record. But it's an animal unlike anything alive today. No head, no tail, no limbs, no eyes, no gut that we can identify. Just a flat, quilted, segmented disc that apparently grew by adding new segments at one end. And there were others. Tribrachidium, which had a bizarre threefold symmetry. Most animals today are either bilaterally symmetric, like you and me, or radially symmetric, like a sea anemone. Threefold symmetry is almost unheard of in modern organisms. Kimberella, which is particularly interesting because it might have been a mollusk-like creature that actually grazed on microbial mats, leaving scratch marks in the sediment. If that interpretation is correct, Kimberella represents one of the earliest animals that actively moved around and fed, rather than just sitting on the seafloor absorbing nutrients. And then there's Sprigina, which some people have suggested might be an early arthropod relative, though that's controversial. The person who really brought the Ediacaran biota to the world's attention was Reg Spriggs, an Australian geologist who described fossils from the Ediacara Hills in South Australia in 1947. But for a long time, the scientific establishment didn't quite know what to do with them. Martin Glessner, also working in Australia, spent decades trying to shoehorn these organisms into existing animal groups. This one's a jellyfish, that one's a worm, this one's a sea pen. And some of those identifications might be partially right. But in the 1980s and 1990s, the German paleontologist Adolf Seilacher proposed something much more radical. He argued that many Ediacaran organisms represented a completely separate experiment in multicellular life, what he called the Vendobionta. In his view, they weren't ancestors of modern animals at all. They were something else entirely, with a unique, quilted body construction that maximized surface area for absorbing nutrients. They had no internal organs because they didn't need them. Every cell was close enough to the surface to exchange gases and nutrients directly with the surrounding water. Think about what that means for a second. If Seilacher was right, and his ideas are still debated, but they've been enormously influential, then the Ediacaran period represents a time when life was running multiple parallel experiments in how to build a large, complex organism. The approach that eventually won out, the one that led to you and me and every animal you've ever seen, was just one of several options on the table. The Vendobiont approach, flat, fractal, quilted, surface area maximizing, was another. And it worked. For tens of millions of years, these organisms thrived in the Ediacaran oceans. They were successful. And then, quite abruptly in geological terms, most of them vanished. Now, why did they disappear? This is one of the great transition points in the history of life, and it connects directly to what comes next. The Cambrian explosion. The Ediacaran biota largely fades from the fossil record by about 541 million years ago, right around the boundary between the Ediacaran period and the Cambrian. And what replaces them is something radically different, something faster, harder, toothier. There are a few hypotheses for why the Ediacarans vanished. One is environmental change, shifts in ocean chemistry, oxygen levels, or nutrient availability that favored different body plans. Another is that the Ediacarans were simply outcompeted or eaten by the new wave of animals that evolved during the Cambrian. If you're a flat, soft, immobile organism sitting on the seafloor absorbing nutrients through your skin, and suddenly the ocean is full of creatures with eyes and jaws and the ability to burrow into sediment, you're in trouble. The evolution of predation may have been the single most transformative event in the history of animal life, and it may have spelled doom for the gentle, passive Ediacarans. And this brings us to the Cambrian explosion itself, which is, honestly, it's one of the most astonishing things in all of Earth's history. In a span of maybe 20 to 25 million years, starting around 541 million years ago, nearly all the major animal body plans that exist today appeared in the fossil record. Arthropods, mollusks, chordates, echinoderms, annelids, the fundamental blueprints for almost every animal phylum we know show up in this relatively narrow window of time. In geological terms, it's like a light switch flipping on. Darwin himself was troubled by it. He devoted an entire section of On the Origin of Species to worrying about the Cambrian explosion, because it seemed to contradict his idea of gradual, incremental change. Where were the ancestors? Where were the transitional forms? We now know that part of the answer is that there were ancestors, the Ediacaran organisms, and probably many soft-bodied creatures that simply didn't fossilize well. The Cambrian explosion wasn't life appearing from nothing. It was life suddenly becoming much more visible in the fossil record because animals started building hard parts, shells, exoskeletons, spines, teeth. And why did they start building hard parts? Almost certainly because of predation. Once some organisms evolved the ability to eat other organisms, there was enormous selective pressure to evolve defenses. Shells, armor, spines, the ability to burrow into sediment to hide, and the predators, in turn, evolved better tools to crack those shells and dig up those burrowers. It's an arms race, and arms races drive rapid evolution. The best window we have into this Cambrian world is the Burgess Shale, a deposit of extraordinarily well-preserved fossils in the Canadian Rockies of British Columbia. Charles Doolittle Walcott, the secretary of the Smithsonian Institution, discovered it in 1909. The story goes that his horse stumbled on a slab of shale on a mountain trail, and when Walcott looked down, he saw the most exquisitely preserved fossils imaginable. Soft tissues, internal organs, eyes, guts, appendages, things that almost never fossilize, all captured in stunning detail in fine-grained mudstone that had been deposited in an underwater mudslide about 508 million years ago. Walcott spent years collecting tens of thousands of specimens, but here's the fascinating part. He largely tried to fit them into existing animal groups, just as Glassner had done with the Ediacarans. It wasn't until the 1960s and 1970s when Harry Whittington and his students, Simon Conway Morris and Derek Briggs at Cambridge, undertook a painstaking re-examination of Walcott's collections, that the true strangeness of the Burgess Shale fauna became apparent. Anomalocaris, the name means strange shrimp, was a meter-long predator with circular, tooth-lined jaws and a pair of grasping appendages at the front of its head. It was the apex predator of its world, and for a long time nobody realized it was a single animal, because different body parts kept getting described as separate organisms. The circular mouth was classified as a jellyfish. The grasping appendages were classified as shrimp. It took decades to put the pieces together. Opabinia had 5 eyes and a flexible proboscis tipped with a claw that it used to grab food and pass it backward to its mouth, which faced backward under its head. When Whittington first presented his reconstruction of Opabinia at a paleontology conference in 1975, the audience reportedly burst out laughing because the animal looked so absurd. 5 eyes, a backward-facing mouth, a nozzle on its face like a vacuum cleaner hose. It sounded like a joke, but it was real. Hallucigenia was originally reconstructed upside down and backward. Its legs were mistaken for spines, and its spines were mistaken for legs. Its name literally means a product of hallucination, because the paleontologist who named it, Simon Conway Morris, thought it looked too bizarre to be real. And then there's Pikaia, a modest little worm-like creature a few centimeters long, easily overlooked among the flashier Burgess Shale animals. But Pikaia had a notochord, a flexible rod running along its back, which makes it one of the earliest known chordates. Chordates, that's our phylum. You, me, every fish, every bird, every mammal, we're all chordates. And our lineage in the Cambrian was a small, unassuming worm-like thing wriggling through the mud, while Anomalocaris ruled the seas. There's something deeply humbling about that. Stephen Jay Gould made the Burgess Shale famous in his 1989 book, Wonderful Life, in which he argued that the Cambrian explosion produced a much wider range of body plans than exists today. That evolution has been a process of pruning, not just diversifying. He suggested that if you could replay the tape of life, the outcome would be completely different each time, because so much depends on contingency, on luck, on which lineages happen to survive mass extinctions. Conway Morris later pushed back hard on this, arguing that convergent evolution, the tendency for natural selection to arrive at similar solutions independently, means that something like intelligence, something like eyes, something like a skeleton, would probably evolve no matter how many times you replayed the tape. It's one of the deepest debates in evolutionary biology, and the Burgess Shale sits right at the heart of it. But here's what I want you to hold on to as you drift along. The Cambrian explosion wasn't just about animals getting bigger or more diverse. It was about a fundamental shift in how ecosystems worked. Before the Cambrian, the ocean floor was covered in thick microbial mats, those same cyanobacterial communities we talked about earlier, the ones that had been oxygenating the atmosphere for over 1 billion years. The Ediacaran organisms mostly lived on top of those mats, or were anchored to them. But Cambrian animals started burrowing. They churned up the sediment, broke apart the mats, mixed oxygen into the seafloor mud. This is called bioturbation, and it completely restructured marine ecosystems. The age of microbial mats as the dominant feature of the ocean floor was over. The age of animals had begun, and the oxygen that made all of this possible, remember, aerobic respiration gives you roughly 18 times more energy per glucose molecule than fermentation. That oxygen had been building in the atmosphere for hundreds of millions of years, through the boring billion, through the Snowball Earth events, through the Ediacaran. Some researchers, like Tim Lenton and others, have argued that there was a second major rise in atmospheric oxygen in the Neoproterozoic, sometimes called the Neoproterozoic oxygenation event, which brought oxygen levels up from maybe a few % of modern levels to something closer to what we'd recognize today. And this second oxygenation may have been both a cause and a consequence of the rise of animals. Animals need oxygen to fuel their active lifestyles, but animals also drive oxygen production through their effects on nutrient cycling and organic carbon burial. It's a feedback loop, and it's the kind of thing that makes Earth system science so fascinating and so maddeningly complex. Nothing happens in isolation. The chemistry of the ocean affects which organisms can survive, and which organisms survive affects the chemistry of the ocean. The composition of the atmosphere shapes evolution, and evolution reshapes the atmosphere. Everything is connected to everything else, across scales of space and time that are almost impossible to hold in your mind all at once. So by the end of the Cambrian, roughly 485 million years ago, the oceans are teeming with complex animal life. The basic body plans are in place. Predators and prey are locked in evolutionary arms races that will continue, in one form or another, for the next 500 million years. But life is still entirely aquatic. The land surface of the Earth at this point is essentially barren. Bare rock, sand, maybe some crusts of hardy microbes and lichens clinging to surfaces near water. No plants, no animals, no soil in any meaningful sense. The continents are vast, empty, windswept expanses of stone. And the story of how life finally crawled out of the water and onto that barren rock, that's a story that starts with something you might not expect. It starts with something green, something humble and quiet and honestly kind of boring to look at. It starts with plants. Now, that might seem obvious when you say it out loud. Of course, plants came first. They had to, right? Animals need something to eat. But the deeper reason is even more fundamental than food. Plants had to come first, because without them, the land was essentially uninhabitable. Think about what bare rock looks like after a rainstorm. The water just runs off. There's nothing to hold it, nothing to slow it down, nothing to break the wind, nothing to buffer temperature swings between scorching days and freezing nights. And crucially, there's no soil. Soil isn't just crushed rock. It's a living matrix of organic matter, minerals, water, air, fungi, bacteria, all woven together into something that can sustain life. And soil doesn't exist without biology. Plants make soil possible. So the colonization of land was, in a very real sense, a terraforming project. Life had to build its own habitat before it could move in. The first pioneers were almost certainly not what we'd recognize as plants today. They were probably something more like algae, green algae specifically, from a group called the charophytes. These are freshwater algae that are the closest living relatives of land plants, and genetic evidence suggests that the common ancestor of all land plants was a charophyte alga living in a shallow freshwater environment, maybe the edge of a pond or a river margin, sometime in the Ordovician period, roughly 470 million years ago, give or take. Some molecular clock estimates push it back even further into the Cambrian, but the oldest unambiguous fossils of land plant spores show up in Ordovician rocks around 470 million years ago in what's now Argentina and Saudi Arabia. These are just spores, tiny, tough-walled reproductive cells, but they have a distinctive structure that marks them as belonging to land plants. The plants themselves were probably small, low-growing things like liverworts or hornworts. No roots, no leaves, no stems in any real sense, just thin green tissue pressed close to damp ground. Here's the thing about moving from water to land. It's an engineering nightmare. In water, you're surrounded by your medium. Water supports your body, delivers nutrients directly to your surfaces, carries away waste, keeps you moist, buffers temperature. On land, you lose all of that in one go. The air is dry, which means you'll desiccate unless you can waterproof yourself. Gravity is suddenly a real problem because air doesn't support your weight the way water does. You need some way to stand upright if you want to get any height at all. You need to transport water from wherever it is in the ground up to your photosynthetic tissues, which means you need internal plumbing. And you need to exchange gases, carbon dioxide in, oxygen out, without losing all your water in the process. Every one of those problems required an evolutionary solution, and the history of plant evolution on land is basically the history of solving them one by one. The earliest land plants solved the desiccation problem the simplest way possible. They stayed small and stayed wet. Liverworts and mosses, the most ancient lineages of land plants still alive today, don't have true roots or vascular tissue. They absorb water directly through their surfaces, which means they can only survive in moist environments, and they can only grow a few centimeters tall at most. But even these tiny, humble organisms were transformative. Their presence began to break down rock surfaces through a combination of chemical weathering, they secrete organic acids, and physical intrusion. Little root-like structures called rhizoids working into cracks. They trapped dust and organic debris. They began, slowly, to build the first true soils, and that changed everything about how water moved across the landscape, how minerals cycled, how the atmosphere was regulated. But the real game changer came later, in the Silurian and especially the Devonian period, roughly 420 to 360 million years ago. This is when vascular plants evolved, plants with internal plumbing. The key innovation was a tissue called xylem, which consists of hollow, reinforced tubes that can carry water from the roots up through the stem to the leaves. Xylem cells are stiffened with a molecule called lignin, and lignin is one of the most important molecules in the history of life on Earth, for reasons that go way beyond just holding plants upright. Lignin is tough, really tough. It's a complex polymer. Imagine a three-dimensional web of carbon rings all cross-linked together, and it's incredibly resistant to decomposition. When early vascular plants evolved lignin, they could grow taller, which was a huge competitive advantage because height means access to more sunlight. The first vascular plants were still small. Cooksonia, one of the earliest we know of, was just a few centimeters tall, basically a forking green stem with no leaves. But the evolutionary trajectory was clear. Once you have vascular tissue and lignin, the race to grow taller begins, and over the course of the Devonian, plants went from ankle height to full-sized trees. By the late Devonian, around 370 million years ago, there were forests, actual forests, with trees like Archaeopteris reaching 30 meters tall. Think about that timescale. In maybe 50 million years, the land went from bare rock with some mossy crusts to towering forests. That's fast, geologically speaking. And those forests changed the planet in ways that are almost hard to overstate. Here's where it gets really interesting. Trees have deep root systems that massively accelerate the chemical weathering of rocks. When silicate minerals weather, they consume carbon dioxide from the atmosphere. It's a chemical reaction where CO2 dissolved in rainwater reacts with silicate rocks, and the carbon eventually ends up locked away in carbonate minerals on the ocean floor. So the spread of deep-rooted forests across the continents during the Devonian drew down enormous amounts of atmospheric CO2. And when you pull CO2 out of the atmosphere, you cool the planet. There's strong evidence that the expansion of forests in the late Devonian contributed to a major glaciation. Ice sheets formed on the southern supercontinent Gondwana. Sea levels dropped, and the climate shifted dramatically. Plants, just by growing and doing what plants do, triggered an ice age. And then there's the lignin problem, which is one of my favorite stories in all of Earth history. Remember how I said lignin is incredibly resistant to decomposition? Well, when those early forests grew and trees died and fell, the lignin in their wood didn't break down. Nothing could eat it. The fungi and microbes that today are responsible for decomposing wood, white rot fungi specifically, hadn't evolved yet, or at least hadn't evolved the enzymes needed to break apart lignin's complex structure. So dead trees just piled up, layer after layer of undecomposed wood, accumulating in swamps and lowland forests, getting buried under sediment, compressed, heated over millions of years. And what do you get when you bury vast quantities of carbon-rich plant material and cook it slowly underground? You get coal. This is the Carboniferous period, roughly 360 to 300 million years ago, and it's named for exactly this reason. It's the age that produced the vast coal deposits that later powered the Industrial Revolution. The coal seams of Pennsylvania, Wales, the Ruhr Valley, Donetsk. They're all Carboniferous in age, and they exist because for tens of millions of years, trees could make lignin, but nothing could unmake it. When white rot fungi finally evolved the necessary enzymes, and there's a beautiful 2012 study by David Hibbett and colleagues that used molecular phylogenetics to pin down the timing, the rate of coal formation dropped dramatically. The era of massive coal deposition essentially ended because biology caught up. Fungi learned to eat wood. It's a perfect example of how the evolution of one group of organisms can reshape the entire planet's geology and chemistry. Meanwhile, animals were making their own tentative moves onto land. The story here is just as dramatic, and it centers on one of the most iconic transitions in all of evolution, the origin of four-legged land vertebrates, the tetrapods, from lobe-finned fish. Now, you've probably seen those classic illustrations of a fish hauling itself out of the water onto a muddy bank. And while that image captures something true about the transition, the real story is more nuanced and more interesting. The key fossils come from the late Devonian, around 375 million years ago, and the most famous of them is Tiktaalik, discovered in 2004 by Neil Shubin, Ted Deschler, and Farish Jenkins on Ellesmere Island in the Canadian Arctic. Shubin has written beautifully about this discovery. They went looking for rocks of exactly the right age and the right depositional environment, shallow freshwater sediments from the late Devonian, and after several grueling field seasons in one of the most remote places on Earth, they found what they were looking for. Tiktaalik is a stunning intermediate form. It has the scales and fins of a fish, but its fins contain bones that correspond to a wrist and fingers. It has a flat head with eyes on top, like a crocodile, and a neck. Fish don't have necks because their skull is fused to their shoulder girdle, but Tiktaalik does. It could do push-ups. It could lift its head and look around. It was a fish that could prop itself up in shallow water and peer over the surface. But Tiktaalik wasn't alone. There's a whole series of transitional forms from this period. Panderichthys, Acanthostega, Ichthyostega, each showing a slightly different combination of fish and tetrapod features. Acanthostega is particularly fascinating because it had fully formed limbs with digits, 8 fingers on each hand, but it also had internal gills and a tail fin. It was a limbed animal that almost certainly still lived primarily in water. The limbs may have evolved not for walking on land at all, but for navigating through dense aquatic vegetation in shallow, swampy environments. Walking came later, almost as an afterthought, a repurposing of equipment that had evolved for a completely different reason. And here's what's beautiful about this transition. It wasn't a single heroic leap from water to land. It was a long, messy, gradual process that played out over millions of years, with different lineages experimenting with different solutions. Some had lungs and gills simultaneously. Some could breathe air, but couldn't support their weight on land. Some had limbs, but probably never left the water. The boundary between fish and tetrapod is genuinely blurry when you look at the fossils, which is exactly what you'd expect from evolution. Nature doesn't draw clean lines, and the descendants of those clumsy, half-aquatic pioneers would eventually give rise to every frog, every salamander, every lizard, every dinosaur, every bird, every mammal, every dog that's ever curled up at someone's feet, every whale that returned to the sea, every human being who's ever lived. All of us trace back to something like Tiktaalik, propping itself up on stubby fins in a Devonian swamp, peering out at a world that was just beginning to turn green. A world that was just beginning to turn green, and would only get greener, wilder, more complex from there. Because once those early tetrapods figured out how to actually support their weight on land, once the limbs got sturdier, the rib cages stronger, the skin better at retaining moisture, a whole new chapter of Earth's story opened up, and it opened fast, at least by geological standards. By the early Carboniferous, around 350 million years ago, you've got amphibians diversifying into all sorts of forms, some of them surprisingly large. But amphibians had a limitation, and it's one you might already know if you've ever watched a frog. They had to go back to the water to reproduce. Their eggs were soft, jelly-coated things that would dry out in minutes if you left them on land. So amphibians were tethered. They could wander, they could hunt, They could bask in the sun. But when it came time to lay eggs, they needed ponds, streams, puddles. They were land animals on a leash. And then something remarkable happened. Somewhere around 310 to 320 million years ago, a group of tetrapods evolved the amniotic egg. And this is one of those innovations that sounds simple but changes absolutely everything. The amniotic egg is basically a self-contained life support system. It's got a hard or leathery shell to prevent water loss, a membrane called the amnion that surrounds the embryo in its own little pool of fluid, another membrane for gas exchange, and a yolk sac for nutrition. Think of it as a private pond, miniaturized and made portable. The embryo develops inside its own tiny aquatic environment, even though the egg itself is sitting on dry land. It's evolution's answer to the water problem. You don't need to go back to the pond if you bring the pond with you. The animals that evolved this trick are called amniotes, and they split fairly early into two great lineages. One lineage, the synapsids, would eventually lead to mammals. To you, lying there right now, listening to this. The other lineage, the sauropsids, would lead to reptiles, dinosaurs, and birds. And for a long time, through the late Carboniferous and into the Permian period, it was actually the synapsids, your ancestors, who dominated the land. Not the dinosaurs. The dinosaurs hadn't shown up yet. The Permian world, from about 299 to 252 million years ago, was ruled by synapsids. Some of them, like Dimetrodon, that's the one with the big sail on its back that everyone thinks is a dinosaur but isn't, were apex predators. Later, synapsids called therapsids became even more mammal-like, with differentiated teeth, upright posture, possibly even whiskers and fur. They were getting closer and closer to what we'd recognize as mammals. The lineage was on a roll, and then the world ended, not metaphorically, almost literally. About 252 million years ago, at the boundary between the Permian and the Triassic, Earth experienced the worst mass extinction in its entire history. It's called the Permian-Triassic extinction, or sometimes just the Great Dying, and that name is earned. Somewhere around 90 to 96% of all marine species went extinct. On land, roughly 70% of terrestrial vertebrate species vanished. This wasn't just a bad century. This was the closest complex life has ever come to being completely wiped out. The cause? The best evidence points to massive volcanic eruptions in what's now Siberia, the Siberian traps. And when I say massive, I mean a volume of lava that covered an area roughly the size of Western Europe, erupting over the course of maybe 1 million years. But it wasn't just the lava. The eruptions punched through enormous deposits of coal and limestone, releasing staggering quantities of carbon dioxide and methane into the atmosphere. The planet warmed. The oceans acidified. Oxygen levels in the deep ocean plummeted, a condition called anoxia, which we talked about way back at the beginning of this story, when Earth's early oceans had no free oxygen at all. In a terrible irony, the Great Dying partially recreated those ancient suffocating conditions. Ocean temperatures may have risen so high in some regions that marine life simply couldn't survive. There's research by people like Justin Penn and Curtis Deutsch suggesting that tropical ocean temperatures during the extinction may have exceeded the thermal tolerance of most marine organisms, essentially cooking them. The recovery was agonizingly slow. For millions of years after the extinction, ecosystems were impoverished, dominated by a handful of disaster species, the weedy generalists that could survive in degraded environments. It took perhaps 10 million years for biodiversity to fully recover. But when it did recover, the world looked very different. The synapsids, your ancestors, the ones who'd been running things, were knocked back hard. They survived, but they were diminished, pushed into the margins, and into the ecological space they'd vacated stepped the archosaurs, the lineage that would give rise to crocodilians, pterosaurs, and dinosaurs. The Triassic, from 252 to 201 million years ago, was the dinosaurs' proving ground. They started small. Some of the earliest dinosaurs, like Eoraptor and Herrerasaurus from Argentina, dating to around 231 million years ago, were modestly sized, maybe a meter or two long. They were fast, bipedal, and probably opportunistic. But they weren't dominant yet. The Triassic was a world shared with lots of other archosaurs, big amphibians, and the surviving synapsids. Dinosaurs were just one group among many. Then, right at the end of the Triassic, around 201 million years ago, another mass extinction struck. The end Triassic extinction. This one was also linked to massive volcanism, this time from the Central Atlantic magmatic province, enormous eruptions associated with the breakup of the supercontinent Pangaea. And once again, the extinction reshuffled the deck. Many of the dinosaurs' competitors were wiped out, and when the dust settled, again, almost literally, the dinosaurs inherited the Earth. And what they did with it is extraordinary. The Jurassic and Cretaceous periods, stretching from 201 million years ago to 66 million years ago, were the age of dinosaurs, and they filled every conceivable ecological niche. Sauropods like Brachiosaurus and Argentinosaurus became the largest land animals that have ever lived. Argentinosaurus may have weighed 70 tons or more, which is almost incomprehensible. Think about what that means structurally. The engineering challenges of building a body that large are immense. The bones have to be strong enough to support the weight, but light enough that the animal can actually move. And sauropods solved this with a system of air sacs running through their bones, making them lighter without sacrificing strength. It's the same basic principle used in modern aircraft construction, hollow structures with internal reinforcement. Birds inherited this system directly from their dinosaur ancestors, which is one of the reasons birds can fly despite having relatively large bodies. And speaking of birds, here's one of the most beautiful stories in all of paleontology. Birds are dinosaurs. Not descended from dinosaurs in some vague, hand-wavy sense. They are literally dinosaurs, in the same way that you are literally a mammal. The lineage that gave rise to modern birds runs straight through the theropod dinosaurs, the group that includes Velociraptor and Tyrannosaurus. And we know this because of feathers. Starting in the 1990s, extraordinary fossils from the Liaoning province in northeastern China began revealing small theropod dinosaurs covered in feathers. Sinosauropteryx, described in 1996, was the first non-avian dinosaur found with clear evidence of feathery integument. Then came Caudipteryx, Microraptor, which had feathers on all four limbs and may have been able to glide, and dozens more. Feathers, it turns out, didn't evolve for flight. They evolved first for insulation, possibly for display, and were only later co-opted for aerodynamics. Flight was another afterthought, another repurposing, just like walking was for those early tetrapods with their limbs built for swimming. Meanwhile, through all of this, those diminished synapsids, your ancestors, were still hanging on. They'd evolved into true mammals by the late Triassic, around 225 million years ago. But for the entire reign of the dinosaurs, mammals were mostly small, mostly nocturnal, mostly hiding. There's a hypothesis well supported by evidence that mammals were essentially forced into nocturnal niches by dinosaur competition, and that this is why most mammals have relatively poor color vision compared to birds and reptiles, but excellent senses of smell and hearing. Your ability to hear the subtle sounds in this recording, to distinguish every nuance of tone and pitch, that's a legacy of 150 million years of hiding from dinosaurs in the dark. You are, in a very real sense, shaped by the age of dinosaurs, even though your ancestors spent most of it trying not to be eaten. And then, 66 million years ago, everything changed again. An asteroid roughly 10 kilometers across struck what is now the Yucatan Peninsula in Mexico, creating the Chicxulub crater. The impact released energy equivalent to billions of nuclear weapons. It triggered tsunamis, wildfires across entire continents, and blasted so much debris into the atmosphere that sunlight was blocked for months, possibly years. Photosynthesis collapsed. Food chains collapsed. The non-avian dinosaurs, along with roughly 75% of all species on Earth, went extinct. This is the end Cretaceous extinction, and it's probably the most famous mass extinction of all, partly because of the dinosaurs and partly because of how it was discovered. Luis and Walter Alvarez, father and son, finding that thin layer of iridium-rich clay at the boundary, a chemical fingerprint of an extraterrestrial impact, published in 1980. It was a detective story written in stone. But here's the thing. The asteroid didn't kill everything. The survivors included some birds, some mammals, crocodilians, turtles, snakes, and many marine organisms. And with the dinosaurs gone, the mammals finally had their chance. Within 10 million years of the extinction, mammals had diversified explosively into bats, whales, horses, primates, rodents. The Cenozoic era, from 66 million years ago to now, is often called the Age of Mammals, and it's the era that produced everything familiar to you. Grasslands appeared for the first time around 25 million years ago, and with them came grazing animals, and with them came the predators that hunted them. The world was assembling itself into something you'd almost recognize. And somewhere in Africa, maybe 7 million years ago, a lineage of primates began walking upright. By 2.5 million years ago, early members of the genus Homo were making stone tools. By 300,000 years ago, Homo sapiens appeared. You, anatomically modern humans, with the same brains, the same bodies, the same capacity for wonder that you're using right now to listen to this story. And what a story it is when you step back and look at it whole. 4.5 billion years, a cloud of gas and dust collapsing under its own gravity, forming a molten ball of rock, getting slammed by another planet, cooling just enough for water to pool on its surface. Then chemistry becoming biology in some warm pond or deep-sea vent. We still aren't sure which, and that's okay. Single cells figuring out photosynthesis, poisoning their own world with oxygen, triggering ice ages that encased the entire planet. Then cells swallowing other cells, becoming something more complex. A billion years of apparent stagnation that was actually quiet preparation. Snowball Earths cracking open, oxygen flooding in, and suddenly, animals. Strange, soft, quilted things on the Ediacaran seafloor, then the Cambrian explosion filling the oceans with every body plan imaginable. Plants creeping onto land, building forests, burying carbon, reshaping the atmosphere. Fish hauling themselves into the shallows on stubby fins. Dinosaurs rising, raining, falling. Mammals emerging from the shadows. Primates standing up. Humans looking at the stars and wondering how it all began. You are made of atoms that were forged in the cores of stars that exploded before the sun was born. The iron in your blood is the same element that sank to Earth's core 4 billion years ago to generate the magnetic field that protects you from solar radiation right now, tonight, as you lie here. The calcium in your bones was once dissolved in ancient oceans, precipitated by organisms that went extinct hundreds of millions of years before anything like you existed. You are not separate from this story. You are the story, still being told, still unfolding. And that's a good thought to carry with you as you drift off. The Earth is still changing. Plates are still moving. Species are still evolving. The story didn't end with us. We're just the latest chapter in something much, much larger. And the same processes that built everything you've heard about tonight, gravity, chemistry, selection, time, they're still running, still shaping, still creating. The Earth is patient. It has all the time in the world. And so, right now, do you. So let that settle over you, like sediment drifting down through a quiet ocean, layer after layer, building something you can't see yet. The story will still be here in the morning. For now, just rest.